Hi everyone, welcome to my channel Srinivasa Biology Tutorial. For this section to discuss NCERT line to line chapter Cell the Unit of Life part 4, that is cell wall. Cell wall. As you may recall, a non-living the rigid structures called as in a cell wall forms an outer covering of the plasma membrane of fungi and plants. So when you see that as in a fungi as well as in some plant groups, the outer the layer is called as what? It is in a cell wall. The cell wall is not only give as in a shape of to the cell to protect cell from as in a mechanical damage and infection, and also it helps to as in a cell to cell interconnections to provide in barrier to their undesirable the microtubules. So it is to give as what? To give as in a particular shape, as well as to give as in a protection, as well as to give as in a the mechanical damage and infection, and also to give as what? It is to also to give as any to interconnections of as in that cell to cell, interconnection of cell to cell. And here you see as in some previous section in that algae, the algae cell wall is made up of what? Cellulose or galactans and then mannans and the mineral like as in a calcium carbonate, while the other plants consist of the cellulose and hemicellulose, pectin and some proteins. Here the cell wall is made up of in a different forms like as in cellulose, galactose and mannans, and minerals like as in calcium carbonate and some plant cellulose, hemicellulose and pectins proteins. Now the cell wall of the primary young plant cell, the wall is capable for as what? Growth. The first one is what? Growth. Now, which as in a generally it is in a diminishes to as cell the mature, and the secondary cell is formed on the inner that towards the membrane side of the cell. So secondary walls also they are formed inner side. So for example, you take as in one particular cell, it is in a outermost layer the cell wall, the inner side also some that layers also to get formed. Okay, like the middle lamella is in the layer mainly is calcium pectate, which is holds that or granule their glues and the different neighbor cells together. So here the middle lamella is made. Here the middle lamella, for example, here they have as in a middle lamella. See here it is in a middle lamella is present. In this middle lamella to get act as what? It is look like a glue. It is help to as in to connecting with as in two cells. The two cells how do they get joined? With help of as in a completely this middle lamella made up of calcium and magnesium pectate. Calcium and magnesium pectate. Now here it holds as in a to glue with different the neighboring cells together. The cell wall is middle lamella is maybe that transferred by as in the plasmodesmatas with connect with as in a cytoplasmic neighbor cells. So the cells how to connect with help of is here as in a the plasmodesmatas connections. Plasmodesmatas connection. Plasmodesmatas connections also to get present here. Now to discuss now in this eukaryotic cell, the major important content is present, that is an endomembrane system. Endomembrane system. While each of the membranes organelles is in a distinct terms, it as in the structure and function. So each and every cell organelle contain as any number of, each every cell they having some cell organelles as present. They have perform as a particular structure as well as in a function. Yes. Now see, the many of these are considered to gather the endomembrane system. Here the many of these are considered together as an endomembrane system because their functions are coordinate. So the number of cell organelles performed as in one common function. Okay, the endomembrane system including as what? Endoplasmic reticulum, that is AR, and Golgi complex and lysosomes and vacuole. So these are called as what? Endomembrane system. These are called as what? Endomembrane system. Now, since these functions, their mitochondria and chloroplast and peroxisomes are not coordinate with above the any components. Here, very important point. This is what the number of times they are asking in some competitive exams, which are not they are completely endomembrane system. Mitochondria, chloroplast, peroxisomes. First one is in mitochondria, and second one is chloroplast and third one is peroxisomes. Third one is peroxisomes. Peroxisome. These are not as in a membrane bound organelles. These are not they considered as in a part of the endomembrane system. Am I right? Correct. Now, so once you discuss about as in some endomembrane system of organelles, the first one is what? Endoplasmic reticulum, simply called as an AR, endoplasmic reticulum. See, you see as an under electron microscopic of the cell, electron microscopic study of the eukaryotic cell, the reveal to as presence of the network or reticulum of the tiny tubule structures scattered on the cytoplasm, that is called as an endoplasmic reticulum. So when you see that as in a one completely in a nearby as in the cytoplasm, some their network like or in a reticulum structures are noticed, those are called as what? In a endoplasmic reticulum. In this endoplasmic reticulum, as in that divides as in a intracellular space into as in a two distinct components. It is in a luminal inside and external lumuli cytoplasm. So they have as a two compartments. One is in inner side and one is in a outer side. Okay. Now the endoplasmic reticulum often show the ribosomes attached to the outer surface. That endoplasmic reticulum bearing as ribosomes, that surface of the structure is called as a rough endoplasmic reticulum, that is in a RAR. See, their outer side they have as in some complete granule like structures are noticed. These are called as what? Ribosomes. These are called as what? Ribosomes. So when the ribosomes is present as an outer surface of the membrane, yes, so that kind of the endoplasmic reticulum is called as what? That is in a rough endoplasmic reticulum. That is in a rough endoplasmic reticulum. Now, in the absence of the ribosomes, there appears as a smooth and they are called as a smooth endoplasmic reticulum, SER. See here, there inside they have as in that granules are absent, the ribosomes are absent. The structures is called as a smooth endoplasmic reticulum. It is what? Smooth endoplasmic reticulum. Now, in this process, the rough endoplasmic reticulum, in this process, here the frequently the rough endoplasmic reticulum absorbed in the cells actively the involved in the protein synthesis. It is a secretion and there the extensive and continuous with as in a outer membrane of the nucleus. Okay, see in this process the rough endoplasmic reticulum the frequently absorbed in that in a clearly active involve the protein synthesis. Okay, and secretion process. Now they are extensive and continuous within the outer membrane of the nucleus. And then next the smooth endoplasmic reticulum, the major the synthesis of the lipids. What? The synthesis of the lipids. And in animal cells the lipid like the steroidal hormones also synthesis as in a the smooth endoplasmic reticulum. Okay, this is the in completely endoplasmic reticulum. And second the endomembrane system, that is the Golgi apparatus. In Cavallo Golgi in 1898, the first observed the densely the stained the reticular structures the nearby nucleus. So nearby nucleus, some the reticular structures are noticed. Those are called as in a Golgi apparatus. When these are then mainly here. These were the latter named as an Golgi bodies after him. So when he discovered as an Golgi apparatus, is that name based on these are called as an Golgi apparatus. They consist of many the fat disc-shaped sacs, the cisternae of the 0.5 micrometers to 1.0 micrometer diameter. So these are completely has any how much they consist of mostly 0.5 to micrometers to 1.0 diameters as present. These are in they're stalked the parallel to each other. So here see that parallelly to get each form. Here the Golgi apparatus to get arranged as in a parallel manner. They varied the number of the cisternae that present the Golgi complex. This Golgi complex cisternae they are concentrically the arranged the nearby the nucleus with a distinct convex cysts or the formation of the cis trans region. So they get forms as in a like the two phases. To get forms as in two phases, like one is in a cis region, it is in a cis region, it is in a trans region. Like the two phases as present, one is in a cis region, another one is in a trans region. Okay, now the convey Here the concave, what is the convex and the concave, the trans of the maturing phase, the cis is in a trans phase, the other organelles is entirely they're different but they are interconnected. So each and every they can interconnected to as any the cell to cell. Now here the Golgi apparatus the principally performed as a function the packing of materials. So this is very important point. Golgi apparatus the major function is what? Packing of material to deliver it to the either the intracellular that targets or secreted outside of the cell. So it is that complete their intracellular targets or secrete for the outside of the cell. Now the materials the packing of the vessels from the endoplasmic reticulum fused with agenesis phase and the Golgi apparatus to move towards the maturing phase. So the endoplasmic reticulum too they move from mesonym to the outside of the maturing phase. Now this explains the Golgi apparatus remain in the close association with as an endoplasmic reticulum. So this Golgi apparatus also they are closely associated with as an endoplasmic reticulum. They are both having nearby. And the number of proteins the synthesized by the ribosomes their endoplasmic reticulum they are modified as an Ncert-Neet the Golgi apparatus before they are replaced the from it is the trans phase. So they complete this they get modified cystine regions in the Golgi apparatus they get completely to get released from as a trans phase. Now the Golgi apparatus is the important site for formation of what? To get synthesis of any glycoproteins as well as in a their glycolipids. Okay, the major function of the Golgi apparatus to formation of glycoproteins and then glycolipids. So this is in a completely in Golgi apparatus. Next one lysosomes. Next one is lysosomes. See, these are the membrane-bound vesicle structures formed the process of packing of Golgi apparatus. So these are completely the packing of in a completely Golgi apparatus. They isolated as in a the lysosomal vesicles has been the formed as in a very the almost the typical the hydrolytic enzymes as present. So they consist of as what? The number of the hydrolytic enzymes as present like as in lipase, protease, carbohydrates because they having as what their optimal pH is present. Acidic pH is present. So this enzyme capable of digesting carbohydrates, proteins, lipids, nucleic acids, everything. So these are completely called as in suicidal bags. Here completely the lysosomes are called as in what? In their suicidal bags. We are called as what? Suicidal bags. Now to discuss vacuole. Next to discuss vacuole. So vacuole is a membrane the bone organized space found in the cytoplasm. So when you take as in any one particular cell, in that cell, they mostly they occupies in a more space for this vacuole. Okay, it contains what? Water sap, and excreted products and other material not like useful for as a cell. So they having as what? Wastewater, the water, sap, excreted products, everything is present on the vacuole. The vacuole is bounded by as in a single membrane is present that is called as tonoplast. So in this enter the here this enter the vacuole is covered with as a one layer is present, one single layer. This single layer is called as what? This is a tonoplast. This is what? Tonoplast is present. Tonoplast is present. Okay, and then here the plant cell, the vacuole can be the occupies 90% of the volume of the cell. So mostly in here 90% of volume to occupies in the vacuole. Now there are plants the tonoplast facilitate as and transport as in the number of ions and to gather to minerals against to concentration gradient to vacuole, hence their concentration of the significantly the higher vacuole to the in a cytoplasm. So your cytoplasm to in the nucleus to as in the cytoplasm the enter the ions or molecules everything to get moved from as in the facilitated means they get moved from as in high concentration to low concentration like the facilitation was takes place. Now you see as in some here vacuoles as present as in different organisms. For example, see here the amoeba has a present as in the contractile vacuole is present. It is important for as an osmoregulation and excretion also. In many cells are like as in a protisters considered as what that the food vacuoles as present. They are formed as in a that Eukaryotic fly but as in a food particles. Okay, like next in the next class. Thank you for watching.